Good morning students. Today we are going to see about the scanning electron microscope. So first of all, the introduction about the electron microscope. So what is electron microscope? It's a scientific instrument that uses a beam of highly energetic electron to examine the object on a very fine scale. So that the examination can yield the information about the topography, morphology, composition and crystallographic informations. The electron microscopy mainly are two types. One is scanning electron microscope, another one is a transmission electron microscope. Here only we see about the scanning electron microscope. So what the main advantage of that electron microscope? That resolving power compared with another microscope. The resolving power is the ability of an imaging devices to see object distinctly that are located at a small angular distances. So here we are using Rayleigh criterions for observing the object at very close. So here we are seeing the some image of scanning electron microscope observing. So first one is the compound microscope image. So second one is the SEM image. It will be clearly we can see the yeast cell. Then E. coli bacteria we have to observe in the compound microscope. It look like pink color. Then SEM image we are observably say the shape of that bacteria. So these are the two same images of the blood cells and then polar grains. So compare with compound microscope, we can use electron microscope for the clear picture representations. So these are the some example for resolving power compared with light microscope and then electron microscope. So what's the main working principle of the scanning electron microscope? Here the scanning electron microscope we are using primary electrons and then secondary electrons. The source is a electron gun in SEM microscope. So the primary electrons is converted into the secondary electrons. That secondary electrons directly observe the object and then it will be imaged on the screen. So these are the scanning electron microscope instrument. So what are the main parts of the SEM? First one is the electron gun. Second one is the electron magnetic lenses to focus the electron beam of that particular object. Then third one is a vacuum pump system. Fourth one is a opening to insert the high vacuum observation chamber. Fifth is a operation panel of the focus and the alignment and magnification tools to positioning of the sample. Then sixth, screen for the menu and image display of the these are the main 7 parts of the scanning electron microscopes. So inside the scanning electron microscope we can see the interior pictures. The electron gun consisting of the cathode and the anode. The condenser lens controls the amount of electron traveling down the column. The objective lens focuses the beam into a spot on the sample. Then a deflection coil helps to deflect the electron beam. Then attract the secondary electrons. Then finally we have to see the same image on the screen. So how to prepare the SEM sample? So this is the very tedious protocol for sample preparation for the SEM. So we need sputter coating and then spider coated in the gold and then 30 millimeter radius aluminum stubs for sample preparation. So first sample coated with a thin layer of conductive material. Then the device called sputter coater. The sample placed on a small chamber, that is a vacuum. Then gold foil is placed in the instrument. Then gas is applied and the electric field is caused the electron to move from the sample. Then again the ions becomes attract to the negatively charged gold foil. Then it will be emit the pictures. This is the sample preparation for the SEM. Then how to working on the SEM? This is the procedure. First the electron can produce the electron beam when tungsten wire is heated by the current. Then the beam is accelerated by the anode. Then beam travels through the electromagnetic field and lenses which focus the downward the sample. Then that mechanism deflects the coil able to guide the beam so that the scan surface of the sample is a rectangular frame. Then finally we have to emit the image on the screen. So this is the working principle of that SEM. Here that red color is the electron beams traveled around the samples. Then we have to see the CRT image formed in the screen. So what are the main advantage and disadvantage of the scanning electron microscopy? Advantages, first of all, it will be gives detailed 3D structure of the particular sample and the topographic imaging and versatile information generated from the different detectors. Then that instrument work very fast. Modern scanning electron microscope generate the data in the digital form. There is no error for that. Then most scanning electron microscope sample require the minimal preparation of the actions. So some of the main advantages are there in the scanning electron microscope. Some of the disadvantages also having. So first one is it's a very expensive and then large. Special training is required to operate the scanning electron microscope. The preparation of the sample is very tedious and then limited to the solid sample and then carry the small risk of the radiation exposure associated with the electron that scatter beneath of the sample surfaces. So what are the main applications of the SEM? We are using all the field of life sciences, mainly the virology for investigation of the virus structure and then cryopreservation. We have to use the surface frozen material for the any of the interior or exterior structure will be changed or not. Then mainly used for the tissue imaging, help to know the cells, how the cells are organized in the networking area. Their organization determine how cells can interact, cell signaling most of the time that we have to using for the investigation of the tissue engineering. Then forensic we are using the presence of the materials and evidence that is otherwise undetectable. Then we are using the microorganisms or any of the sample, we have to view the surface area of the sample, mainly the anatomical picture of the insect, worms, spores and other organic structures. Thank you.